Hello students, I am Savita More from Nasik Road College, Nasik. Today we are going to study third topic, that is inheritance and variation, and subtopic is genetic basis of inheritance. This is our first lecture. In previous classes you have studied about genetics. Genetics is study of heredity and variation. Heredity means inheritance. Certain characters from our parents are transmitted in us. It is due to heredity, or we call it as inheritance. So, heredity is transmission of characters from one generation to the next generation, while variation is transmission of certain dissimilar or certain different characters from parents to their offsprings. So, genetics is study of heredity and variation. You know, like begets like. For example, dog will give birth to puppy only, while cutting of rose plant when cultivated will give rise to rose plant only. So, here characters are transmitted from parents to their offsprings. Gregor Mendel, son of the peasant farmer, was born in Moravia in 1822. Gregor Mendel first gave the accurate explanation for the mechanism of inheritance by using hybridization technique. This hybridization technique you are going to study in detail in chapter 11, that is enhancement of food production. Here in hybridization technique, pollen grains from anther they are collected and artificially dusted over the stigma of emasculated flower or male flower. So emasculated flower means when anthers are removed we call it as male flower or emasculated flower. And then process of pollination is followed by seed formation and fruit formation. The seed thus obtained represents the hybrid generation, and Mendel during his time he had performed this experiment which today we call it as hybridization technique. Mendel is known as father of genetics. Mendel postulated the principles of heredity, which then became the fundamental laws of heredity. Now, see the reasons for Mendel's success. His experiments were carefully planned and involved large samples. He carefully recorded the number of plants of each type and expressed his results as ratios. In the pea plant, 7 different characters can be easily recognized. They are controlled by single factor each. 7 different contrasting characters are easily recognized in pea plant. That is height of the stem, tall and dark. These are the contrasting characters. Then color of the seed, yellow and green. This is also example of contrasting character. So for this reason, Mendel selected this plant and he also got successful. The factors are located on the separate chromosome and they are transmitted from generation to generation. Here factor means gene. During the time of Mendel, microscopes were not invented or genes were not discovered. So he used to call as this unit of heredity as factor. Now see different genetic terminology. Character. It is a specific feature of an organism. Example, height of the stem. In previous chart you had studied 7 different characters. So one of the example is height of the stem, then color of the flower, color of the seed, color of the pod, position of the flower, all these are the characters. Trait, an inherited character and its detectable variant. For example, tall and dwarf, round and wrinkled, axillary and terminal. So these characters are inherited and they are detectable variant. So this is called as trait. Factor. It is unit of heredity, a particle present in the organism which is responsible for the inheritance and expression of a character. During the time of Mendel, microscopes were not invented or genes were not discovered. So he used to call this unit of heredity as factor. Then gene. Gene, it is a particular segment of a DNA which is responsible for the inheritance and expression of that character. So what is gene? Gene, it is a particular segment of a DNA which is responsible for the inheritance and expression of that character. See the next term, alleles or allelomorphs. The two or more alternative forms of a given gene are called alleles of each other, and they occupy the identical loci or position on homologous chromosomes. For example, capital T, capital T. These are alleles of each other to show tall character. Small t, these are also alleles of each other and it is used to denote dwarfness. Capital T, small t for hybrid tall plants. Next term, dominant. It is an allele that expresses its trait even in the presence of an alternative allele that is in heterozygous condition only. Alternatively, the allele that expresses in F1 generation is called dominant. Example, capital T, small t, that is hybrid tall plants. While recessive, this allele is not expressed in the presence of an alternative allele that is in heterozygous condition. While dominant allele, they are expressed in the presence of an alternative allele. Recessive does not express itself in the presence of alternative allele. Dominant is expressed in F1 generation, while recessive is not expressed in F1 generation. Example of recessive is small t, that is dwarf. Phenotype. The external appearance of an individual for any trait is called phenotype of that trait. For example, height of the plant tall, color of the seed yellow, color of the pod green. So we can see or we can observe its external, we can tell the character on the basis of its external appearance. It is called as phenotype. Genotype. Genetic constitution of an organism with respect to a particular trait. It is representation of genetic constitution of an individual with respect to single character or set of characters. For example, tall pea plant have genotype capital T or capital T small t. Homozygous. An individual possessing identical alleles for a particular trait or character is called homozygous or pure for that trait. For example, tall, pure tall with capital T and dwarf with small t. So here you see both the alleles are similar. That's why the term used is homozygous. Homo means same, while heterozygous. Hetero means different. An individual possessing contrasting allele for a particular trait is called heterozygous. Example F1 hybrid, that is capital T small t. Pure line, an individual or group of individuals which are true breeding, means they are pure for one or more traits constitute pure line. Monohybrid, it is heterozygous for one trait and is produced from cross between two pure parents differing in single pair of contrasting character. So mono means single. When single character is used for crossing, it is called as monohybrid. F1 generation. It refers to the first filial generation. Filial means son. So it is the first generation after crossing. It consists of all offsprings produced from a parental cross. F2 generation, the second filial generation produced by selfing of F1 generation offsprings, is called second filial generation. Example, progeny. Progeny or offsprings produced from cross between two F1 individuals. Example, capital T small t into capital is crossed with capital T small t. Here selfing is done between the individuals obtained from F1 generation. Homologous chromosome. The morphologically, genetically and structurally identical chromosomes present in a diploid cell. Diploid cell means a cell having two sets of chromosomes are called as diploid cell. So, homologous chromosomes are the chromosomes which are morphologically, genetically and structurally identical to each other. Phenotypic ratio. It is the ratio of the offsprings produced in F2 and subsequent generation with respect to their physical appearance. For example, 3 tall 3 is to 1, that is 3 in the proportion of 3 tall plants are found, while in proportion of 1 dwarf plants are found. So the monohybrid cross phenotypic ratio is 3 is to 1. Genotypic ratio. It is the ratio of the offsprings produced in F2 and subsequent generation with respect to their genetic constitution, that is 1 is to 2 is to 1, found in monohybrid cross. So capital T in the proportion of 1, capital T small t in the proportion of 2 and small t in the proportion of 1. Now we will study monohybrid cross. A cross between parents differing in one heritable trait is called monohybrid cross. So here it is a cross between two pure or homozygous parents in which inheritance pattern of only one pair of contrasting character is studied. It is called as monohybrid cross. Mendel first selected pure tall pea plant as a female parent and pure dwarf pea plant as male parent. He confirmed that they are true breeding or they are pure by selfing them or by self-pollinating them for at least 3 generations, and then he obtained these pure plants. He then performed artificial cross, collected seeds, sowed the seeds and waited for the result. He was expecting that the offsprings, that is F1 hybrids, will either be of intermediate height or 50% will be tall and 50% will be dwarf. But to his surprise, he found that all were tall. He started thinking, where the dwarfness has gone, why and how it got eliminated, is it eliminated forever, are the tall offspring similar to the parent tall? To find out answers, he proceeded further. In this given chart, you see phenotype, genotype, gametes, etc. They are written one below the other. While towards other side, tall, dwarf, capital T, small t, all these are written. So phenotype, we had already seen the different terms. So phenotype, that is on the basis of external appearance. Tall we can see, dwarf plants we can see. Genotype, that is on the basis of genetic constitution. So capital T and small t, they are written below tall and dwarf respectively. Then below that, gametes. Capital T will give rise to only one type of gamete because it is homozygous, while small t will give rise to only single type of gamete, that is small t. Then after crossing, we had seen hybridization technique. In this hybridization, Mendel cross-pollinated artificially, that is male plant. From the male plant, pollen grains are collected and they are dusted on the stigma of the female parent. So you are going to study all these techniques or all the steps of hybridization in chapter 11. Here Mendel performed this hybridization technique by using pure plants. Then after artificial crossing, he obtained all the plants in F1 generation, tall plants. But these tall plants were hybrid tall. Here genotype is mentioned of hybrid tall, that is capital T, small t. Then as this hybrid tall is heterozygous in condition, two types of gametes are obtained, that is capital T, small t, containing male gametes and female gametes. So here, as this F1 hybrid is heterozygous, two types of gametes were obtained, that is capital T containing male gametes and female gamete, and capital T, small t containing male gamete and female gamete. For obtaining F2 generation, Mendel did selfing of these F1 plants, F1 generation plants, that is self-pollinated. Self-pollination is done in this F1 hybrid plants. Then he obtained F2 plants. F2 plants shows three different genotypes. And these F2 plants are placed in this Punnett square, or it is also called as checkerboard. In this checkerboard you will see different columns. 4 columns are there. Why these 4 columns are obtained? Because two types of gametes are obtained, that is male gametes and female gametes containing capital T and small t. They are placed in this column, male gametes and female gametes. Now see, when male gamete containing capital T fuses with the female gamete containing capital T, here individual obtained will be capital T, that is homozygous tall or pure tall. Then male gamete with small t when fuses with the female gamete containing capital T allele, here plants obtained will be hybrid tall or heterozygous tall. Then male gamete containing capital T and small t, here individual obtained will be hybrid tall. And in the last column in the corner you see homozygous dwarf plant, that is small t. Why? Because here male gametes and female gamete, both the gamete contain similar allele, that is small t and small t. Phenotypic ratio is 3:1. That is tall plant obtained are of 75%, while 25% plant obtained are dwarf plant. So on the basis of external appearance, when we find out the ratio, 3 is to 1. While on the basis of genetic constitution, when we write the ratio, it will be capital T in the proportion of 1, capital T small t, that is hybrid tall plants, in the proportion of 2, that is 50% plants will be hybrid tall, 25% plant will be dwarf plant. 25% pure tall, 50% hybrid tall, and 25% dwarf plants. So the genotypic ratio is 1 is to 2 is to 1. On the basis of monohybrid cross, we can explain law of dominance. In this law of dominance, here each character is controlled by a pair of factor, and when it is a pair of dissimilar factor, only one factor is expressed. Thus one is dominant and other is recessive. You find in F1 generation all plants obtained were hybrid tall, and it consists of genotype capital T small t. Capital T is dominant, small t is recessive. So in F1 generation all plants obtained are tall, and as this tall plant it is a dominant character. Here we can conclude from this law of dominance, or Mendel explained law of dominance on the basis of this monohybrid cross. On basis of dihybrid cross also law of dominance can be explained. Next law of inheritance. First law is law of dominance, and second law of inheritance is law of segregation or law of purity of gametes. Here in the law of purity of gametes, members of allelic pair in a hybrid remain together without mixing with each other and separate or segregate during gamete formation. Thus gametes receive only one of the two factors. We had seen only one factor is received in these gametes. Thus gametes receive only one or one of the two factors and are pure for given traits. Therefore it is also known as law of purity of gametes.